Alright, hey guys, what's going on? So today we're going to be talking about more biophysics. So in the last video we talked about a lot of things, right? We talked about DNA. We sort of got that out of the way because this is a lot of this is going to be introductory kind of stuff. Now we're going to be talking about amino acids, okay? In the last video we talked about a central mapping problem. The problem was, how can we go from a large degree of information to finite structural proteins that can actually do things? And we sort of addressed that question, not in too much depth, but in this video we are going to, now that we talked about nucleic acids in that video, we're going to talk about amino acids in this video. And amino acids are the building blocks of proteins. And then we're going to get more into details later. So without further ado, again, make sure to hit that like and subscribe button. Make sure to go into my Patreon page as well. And let's get right into it. So today we're talking about protein structure and function. Specifically, we're going to be talking about the sequence of proteins today. Proteins are made up of amino acids, right? And there's 20 different types of amino acids. These amino acids are, I'm just going to list them out, right? Because this is something that you just really have to memorize, okay? And I'll give you some tips on how I memorize this kind of stuff as well. And you can go onto my Patreon page where I am making summary sheets so that you can take a look at these proteins, take a look at these amino acids, and start memorizing them so that you can ace your exams, get into medical school, or whatever you're using these videos for. So arginine is, so we have arginine, histidine, lysine. These are all the positive amino acids. So when we say positive, negative, and all this kind of stuff, we mean that at physiological pH, right? Physiological pH, that is around 7. And if you don't know what pH is, that's a topic for chemistry, which is something that I'm very interested in going over as well. But at physiological pH, we're asking the question: is the amino acid basic, or is it acidic? Is it basic or is it acidic? What exactly is happening on the molecular scale when we're talking about physiological pH, which is around, again, 7. Ranges between 7 and 8, but around 7. What's going on here? So physiological pH, arginine, histidine, and lysine are positively charged. Okay, positively charged, simply put, means that there's an extra bond on that atom. All right, think of it like that. Think of it as positive charge. There's an extra bond on that atom that you're looking at, and that atom, you could literally think of it as an extra bond on the atom, right? And so, so in the case here, we have arginine. Okay, arginine is attached to 4 things here. It's typically attached to only 2, right? You typically have NH2, right? So we have 3 things, right? Attached to 3. So there's 1 thing, and then there's 2 of these guys, right? In this case, there's 2 bonds, and there's 2 bonds here, right? So again, extra bond, okay? You can literally think about it like that. And so I'll erase that really quick. So histidine, one of these nitrogens, is going to be bonded to a hydrogen. All right, and so that we're going to have a positive charge on that nitrogen. So these are the positively charged guys at physiological pH. Right, that's these guys right here. The negative guys are these acids. Okay, again, acids, think of them as negative. They're naturally negative at physiological pH. So we have aspartic acid, glutamic acid. These guys look like this. Negative means 1 less bond. Right? So oxygen typically has 2 bonds to it that makes it neutral. So if there's 1 less bond, it's going to be negatively charged. Okay, that's the really easy way to think about this. So if you want to get into the weeds, 1 less bond means there's extra electrons on the atom. Extra electrons on the atom makes it negatively charged. Okay, And so, but that's again topics for chemistry. And so we have positive, negative charges. These are the amino. There's 5 of these guys that have charge on them. Okay. Then there's uncharged guys. So these uncharged guys, there is going to be. So we have serine, threonine, asparagine, and glutamine. And I'm realizing right now that a few things got erased. So glutamine is Q, and asparagine is ASN, and that I believe is R. Glutamine is going to be GLN, if I remember that correctly. And these are all the uncharged guys, right? You notice all these backbones are the same. All these backbones are the same. The one thing that's changing are these guys right here, these green things right here. These are the these are the part of the amino acids that make them unique. Okay. And then we have the hydrophobic guys. So the hydrophobic guys, there's going to be a lot of these, right? So these are all of them. So we have alanine, valine, isoleucine, and leucine. Right? These are all, they just have carbons on them. And then we have methionine, phenylalanine, tyrosine, and tryptophan. Tryptophan's particularly interesting because we have this thing will actually fluoresce. There's tryptophan fluorescence, if that's something that you're not aware of. We're going to go into a little bit, but this thing can actually fluoresce, and you can actually determine the concentration of tryptophan in a protein or the amount of tryptophan in a protein just by shining UV light on it. And we'll get into that later. Then we have special cases, right? So the special cases are these guys. So where we have glycine, right here, glycine's interesting because it doesn't have an extra group on it. It's just hydrogens. And then we have cysteine. So cysteine here is has this guy right here, and proline's quite interesting also because it's R group. I call it R group. I haven't really introduced what an R group is, but the R group is these green side chains, and these green side chain for proline is attached to the amino end of the peptide. Right. When I say peptide, I mean the amino acids. Right. So the backbone of the amino acids. The amino acids themselves make up peptide. They make peptides, a string of peptides. And so, okay, so now let's take a look at these peptides. We can make peptide bonds, and we can deconstruct peptide bonds. If we want to make peptide bonds, we just add them together. Right? We don't. What you get out of that is a new peptide bond and water. So dehydration leads to a peptide bond. Okay, and that's this process right here, right? So this O and these two H's will create the water, and we have this formation of a new peptide bond. And we're going to have to rotate a few things so that things remain sort of sterically unhinged from one another. And this is a depiction of what that looks like. And then we have these amino acids can be in two different isomeric forms as well. We have L isomers and D isomers, where D isomers are going to be the most common, right? So if you ever hear or read a paper that says, say, D-serine on it, I've read quite a few papers on this kind of stuff, D-serine. Well, that's what this is talking about. It's talking about this chiral form of, or this isomer of the peptide, right? So you can have L-serine, which is going to look like this. So serine, in which this is coming out of, this R group is coming out of the plane, and then D-serine. These are two different things, right? Because if you reflect this guy onto this guy, well, you can get the R aligned with this R, but when you do that, this side of the peptide is not going to map onto the side of the peptide. It's going to be on this side. You can just picture sort of flipping it across this guy here, from here to here, and you see that these two guys are not actually the same thing. That means they're chiral. So that means that this carbon here is chiral. Okay. So out of the 20 amino acids, 7 of them have ionizable side chains. So if we're just talking about the carboxyl side here, this is what that is going to look like. Again, at physiological pH, we're going to have half of these and half of these. If the pKa, or sorry, not at physiological pH, right? If the pKa, if the pH is equal to the pKa, then we have half of these guys and half of these guys. Okay, if we go above or below, then we're going to favor one of these, either this side or this side. Okay, so if we go below the pKa, if the pH goes below the pKa, then we're going to get a situation in which we favor, again, one side being one of the two sides, right? So if the pH, and we'll get into this a little bit more actually. So if we get into, and I think I'm going to have this be the content of the next video, where going above or below the pKa of a particular protein or of a particular amino acid, that's going to tell us something about which side of the equilibrium we're on here. Okay, and then we have this for the amino group as well. Again, we have negative charge means less things are bonded. Positive charge means more things are bonded. Okay, and then here are those 7 groups, right? So histidine, the pKa is going to be around 6, and this is what's going on there, right? So at a pH of 6, we have half of these and half of these in solution. At a pH of 8.3 for cysteine, we have these guys. Okay, at a pH of 10.8, we have this situation going on. pH of 10.9, we have a situation of this going on for tyrosine. At 12.5, arginine's looking like this situation. And at a pH of 4.1, we have aspartic acid and glutamic acid looking like these guys. Okay, and so this is all fun and good. What we're going to do in the next video is we're going to go look at a few examples of how this all works. We can imagine, say, we're at a pH of 7, right? So if we're at a pH of 7, well, that's between here and here. Okay, so we can ask the question, what exactly are the cysteines doing and the histidines doing at a pH of 7? All right, so at a pH of 7, we're going to favor one side of these two guys, and at a pH of 8 we're going to favor one side here, and we're going to favor the opposite side here. Okay, and so that's going to be the topic for the next video, where we just want to do some example calculations of what's going on when we talk about acids and bases at particular pHs, right? Because if you're doing this in the lab, which is what I do all the time, then it'd be good to know what exactly is going on, sort of on a molecular level, when you, say, purify a protein at a pH of 7, or when you purify a protein at a pH of 8. Right? We usually do it at a pH of 8 in the lab. That's typical standard procedure in the lab, and so we want to take a look at what's actually going on the molecular level for these proteins. That's what I got for this video. It's a fairly short video. In this video we talked about all the amino acids, and okay, so we've covered amino acids and nucleic acids, right? So let's see, I'll make a new page here Well, what have we covered so far? We've covered amino acids and nucleic acids. Okay. These are acids, okay? And we had this, what I had initially was this mapping, right? So from nucleic acid space to amino acid space, we map from here to here. And this mapping, this was a multifaceted mapping, right? So we can map in this space to here. I'll make this yellow because these are different points. We can have a mapping here. This seems quite arbitrary, but the point here again is that we can actually modify this guy. We can actually modify the sequence in this space. And this space here is that all the RNA modifications, slicing or splicing, post-translational modifications, I'll just abbreviate it as PTM. So all of these guys are the functions. All of these guys here are the functions that take us from information space to function space. And what we're going to find, we're going to do a lot of physics, we're going to understand on the molecular level how all this happens. Okay. It'll be very interesting, very, very fun. We're going to take a look at all the structural biology that plays a role in this as well, which is going to be quite fun and interesting. But with that being said, make sure to hit that like and subscribe button, and I will see you guys in the next video. Bye.